This is the second session of the chapter biomolecules. In my previous session, I discussed about the biomacromolecule. There are four actually. They are found in acid insoluble fraction. If you remember trichloroacetic acid method, there are two fractions in that acid insoluble fraction. In that we can find four different types of biomacromolecules. The first one, the protein, we have discussed the structure as well as the functions of proteins in our previous video session. The second one, second biomacromolecule, that is polysaccharides, I'm going to discuss in this session. Next one is lipid, which is already discussed. Next one is nucleic acid. I'm going to discuss about the structure of DNA in my next session. Okay, so we'll discuss the next biomacromolecule. Let me just write the heading, biomacromolecule. What is biomacromolecule? The molecule or biomolecule which weighs or which has biomolecular weight more than 1000 daltons, Okay, so this is what you need to remember all the time, very important concepts. Okay, so here the second, I'll just write second biomacromolecule, that is polysaccharides. Okay, so what is this polysaccharides or saccharides? Saccharides are nothing but sugar. They are sugars. Simple, right? Okay, we write sugar. Can you just list out some simple sugars? Glucose. Okay, so we write glucose is a simple sugar. Next one, the sweetest one, that is fructose. Yes. Okay, or even this one, the ribose. Where do you see ribose? Sugar in DNA. Yes. Okay, so these are some examples, but I'm talking about polysaccharide. Poly means many. So this particular carbohydrate is made up of many individual sugar units, like glucose, 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 or glucose, fructose, like that. Got it? So that is polysaccharide. Okay, so we just write a very important point, that is where do you see them? It is found in acid insoluble fraction. I hope you can make out my handwriting here. It's very difficult to get a proper grip and position while writing. Okay, next is we just go with the next point, acid insoluble fraction. Okay, so ultimately we just write this point, many monosaccharides, that is simple sugars, are linked. For example, let me write glucose here, glucose here, glucose here, glucose here, like this. Okay, to make a long chain which is called as polysaccharide. This is it, simple. Okay, so many monosaccharide units are linked one another like this. There is a bond, actually I'll tell you the name of it. Okay, ultimately make a long chain, that is called as long chain, or it is also called as polymer. Okay, so this is simple. Now I just want to tell you that polysaccharides are of two types. Okay, so write polysaccharides are of two types. What is the type? The first one is homopolymer. What is this homopolymer? I'll tell you. Next is hetero. Hetero means different, homo means same. Heteropolymer, homopolymer. You get a question actually for your competitive. Homopolymer examples, heteropolymer examples. So this is very important now. Okay, so homopolymers. Homopolymers means it is made up of similar type of monosaccharides. A long chain which is made up of similar type of monosaccharides, like glucose, 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 glucose. You don't see any other different monosaccharide here. Got it? So that is homopolymer. Okay, so made up of same or similar type of monosaccharides. And this one, the heteropolymer, what should I write here? It is containing in a chain of polysaccharide containing different types of monosaccharides. That's it. So this is the simple thing to understand here. Can we write the example for homopolymer here? Okay, so this is also very important. Now let me just take black ink now and let me write here the example for homopolymer here. Okay, this is very important. Cellulose. Cellulose is made up of many glucose units. Many glucose ultimately makes a cellulose. You know about cellulose. It is one of the important component in cell wall of the plant. Yes, right? Yes. Okay, cellulose is one example. Next one is inulin. Okay, so I'll talk about this later. Inulin is another one that is also a homopolymer. Next one is starch. I think you are familiar with this. What is starch? It is the storehouse of energy in plants, starch. Next one is the glycogen. Yes, it is also made up of many glucose units. Glycogen is the storehouse of energy in animals. Yes, in our liver we have glycogen. Okay, so these are the 4 examples for homopolymer. Remember, you get a question: polysaccharide, which is a homopolymer, cellulose, inulin, starch, glycogen. Done. Okay, so next one is a heteropolymer. Okay, so this one, let me just give you the example. Glucosamine is one example. Glucosamine is one example here for heteropolymer. I just write one more. N-acetyl galactosamine is the second example. Okay, the next one, the very important one, is the chitin. What is chitin? You remember, it is the main component in the exoskeleton of arthropod. Chitin, remember this. So glucosamine, N-acetyl galactosamine and chitin are very good example for heteropolymer. That means these are made up of different types of monosaccharide units. Done. Okay, so now let us talk about the linkage. This is very important. Please note this point. Okay, linkage. What kind of bond is there in a polysaccharide? Bond. What type of bond? You remember, amino acids are linked by peptide bond. Yes, which is nothing but a type of covalent bond. Yes. Okay, so linkage, that is the bond in a chain of polysaccharide or homopolymer or heteropolymer chain. You see a type of 1,4 right here, that is glycosidic linkage. So this is very, very important. Okay, so what kind of bond is seen? For example, let us just draw a homopolymer, that is glucose here, and I'm going to draw one more glucose, because homopolymer means similar type of monosaccharides. Okay, so ultimately making a long chain. This is now polysaccharide. Let us simply assume that this is cellulose now. Cellulose also mainly made up of many glucose units. Okay, so this one is the linkage. What is it? 1,4 glycosidic linkage. What is this 1,4 glycosidic linkage? Why I did write 1 and 4 here? Okay, just I'll show you that. For example, if I draw a cellulose, okay, so this is now cellulose. This is 1 glucose. 1 glucose. Okay, so I'm going to draw one more glucose here. I'm going to draw one more glucose here. I'm going to draw one more glucose here. See, many are there, actually a long chain, correct? Okay, now the bond. See, this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon in the glucose. Same here, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, like that. So it is understood. So 1, 2, 3, 4, 5, 6. The linkage is seen between the first carbon of the first glucose. See, okay, first carbon of the first glucose with the fourth carbon of the second glucose. So this is the first glucose and this is the second glucose, this is the third glucose, this is the fourth glucose. It goes on like that. So the linkage is like this. See, so there is a linkage here. This is 1,4 glycosidic linkage or glycosidic bond. Got it now? You got it right? Okay, so again 1,4 1,4. So it goes like that. So this is why we have written 1,4 glycosidic linkage. Yes, so hope you understood. So this is what? This is common to all types of polysaccharide. The linkage 1 and 4. That's it. Okay. So chalo. Next one, let me just tell you some examples for this polysaccharide. That is, we go with a simple homopolymer. I just write cellulose here. I have already told you that it is a type of homopolymer made up of similar type of monosaccharides, that is glucose. Okay, so glucose. Cellulose structure is not helical. Please note this point. It is not helical in structure. So it's a straight, linear. So cellulose is a linear structure. It has got branches. For example, if I draw it in a simple line-like structure, it has got branches. See here, see the branches. This is one chain and this is another chain which is linked actually, and another chain which is linked to this chain. So it's actually kind of like branch or branched structure. Okay, but it is linear, simple. Only glucose is there. Other than that, nothing. Which is actually linked by 1,4 glycosidic linkage. Done. Okay, so it is not helical, and as a result, it cannot hold. It can't hold iodine molecule, I2 molecules. So if I take this cellulose and treat with iodine molecule, the cellulose will not react with the iodine molecule and the solution will not turn to blue. This is what important. Whenever any molecule reacts with iodine, the solution should be blue. Okay, the inference should be blue, but here we are not getting blue. Why? Because the cellulose cannot hold to iodine molecules because it is not helical. The structure will not allow it to hold iodine molecule. So got it? Okay, so let me just give you one more example, that is inulin. So this is another polysaccharide which is also a type of homopolymer, inulin. Okay, so sorry, not inulin, I'm sorry, actually this I should change it. Let me just go with this itself. I'll talk about inulin later. Starch. Okay, starch. So we know that starch is nothing but the storehouse of energy, where in plants. Okay, and the structure of starch, which is actually bit of helical, kind of like this. Okay, so because of the structure which allows to hold iodine molecules, when I treat this starch with iodine molecule, it can hold the iodine and the entire solution becomes bluish in color. So this is what we need to remember. So I'm going to write why it can react with iodine molecules, the starch, because the structure of the starch is helical. Please note this point. Whereas cellulose cannot hold because it is not helical. It cannot hold to iodine. Done? Are you getting my point now? Yes. Okay. So now let me just write one more point. I think I have already mentioned it, but some simple functions I just want to write. Functions or economic or commercial uses of these polysaccharides. Okay, so first one, we just write cellulose. We know that cellulose is the important component in all plant cell walls. Okay, so plant, I just write here plant cell walls. Okay, and also we know that because of the presence of cellulose, we can make this, what is this, paper. Yes, okay, I write. So paper industry, because of cellulose, we can manufacture a lot of papers from the pulp, plant pulp. Yes, okay, because of that this paper is rigid. Okay, so this thing. Next one, let me just give you one more, that is the starch. I have already told you, that is storehouse of energy in plants. Okay, whereas glycogen, that is storehouse of energy in animals. Okay, next one, let me just give you one more example, that is I don't remember. Chitin. Yes, I have already explained it. Chitin is the exoskeleton, the component that is exoskeleton in arthropods, insects. Okay, it makes exoskeleton in arthropods. That's all about it. That's very simple. If you want, I can add one more point about inulin, not insulin. Insulin is a hormone. Inulin is polysaccharide. Okay, so this inulin, which is made up of many fructose monosaccharides. So it is homopolymer or heteropolymer. Yes, you are right, it is homopolymer. Easy now? Okay, so that's all about from this session, a small session actually. So that is polysaccharide. So we understood the protein, the first biomacromolecule, and second one is polysaccharide. Okay, I'm not going to discuss the third biomacromolecule, that is lipid, which is already done in the class. Next one, in the next session, I'm going to discuss about the fourth and very important biomacromolecule, that is nucleic acids. Okay, we discussed that. Hope you understood this entire session. Thank you very much. Stay safe.